Welcome everybody to this afternoon's Water Resource Forum. We're part of the St. Croix River Association. My name is Don Hansen. I'm the chair of the Water Resource Committee. The forum is part of a program that was started by the St. Croix River Association, and we hope to have many more in the future. Today we have Dr. Richard Kiesling speaking to us from the U.S. Geological Survey. Richard holds a Bachelor's of Science in Biology from the University of Minnesota, and he did his doctorate work at the University of Michigan, specializing in limnology and community ecology. He's currently the Lakes and Water Quality Specialist at the U.S. Geological Survey in Mounds View. It has done extensive work on the St. Croix River and its tributaries. So, welcome, Richard.
Thanks, Don. There are a lot of familiar faces in the audience here, and welcome to people who are watching this on YouTube. I want to keep this informal. It's a brown bag, and just give you a little idea of where we're going to go. Don asked me to sort of review what USGS has been doing in the St. Croix, and it's quite a large list of activities that go beyond just what we're doing out of the Minnesota Water Science Center. It involves the Wisconsin Water Science Center out of Middleton, Wisconsin, but there's also a field office in Rice Lake, Wisconsin, and they've been involved in some of our work in St. Croix, and they're doing some work on the bridge mitigation projects, which I'll be talking about. And also there's the Upper Midwest Environmental Science Center near La Crosse, Wisconsin, and on Alaska, and they've done quite a bit of work, or are involved in a fair amount of work in the basin as well. So I'm just going to spend a little bit of time going over some of those projects, just introducing you to the wide array of activities that USGS is involved in. And I just wanted to start out by acknowledging that there are a number of science centers in the area and actually across the country. And we also have some researchers, hydrologists, coming from across the country to work in the basin as well. There we go. So just going through a list of things that we are involved in. Many of you are aware of the USGS through our stream gauging activities. Currently, we have the Minnesota Water Science Center running 4 stream gauges. There's an example here showing a picture of the Snake River gauge near Pine City. You can, from the red triangles, those were active gauges recently. At least one of them, I think, is now discontinued. But the Wisconsin Water Science Center is running 10 or so gauges. One of the things I wanted to point out is these current gauges here on the Apple, Willow, and Kinnickinnick rivers are involved in a project that's funded through the Wisconsin DNR from bridge mitigation funds to track sediment and nutrient loading on those tributaries, and I'll talk a little bit more about that in a few minutes. And those load estimates are actually going into another big project that I'm involved in, that we're doing in the Minnesota Water Science Center, which is developing a water quality model, a dynamic water quality model for the lower St. Croix River, the part that we commonly refer to as Lake St. Croix. All through the presentation, I'm going to have references to our web page URLs so that you can access information and find out more about a topic as it goes by. These first few slides are part of a poster that we presented, or that Jeff Zigwide put together, and he presented at the River Falls annual meeting on the St. Croix, and we had some copies in back for people to pick up, and I believe we're going to make that available on the River Association's website. So that's also where you can find these web links. Okay, so I talked about the Upper Midwest Environmental Science Center in La Crosse. They have been doing quite a bit of organismal biological work in the river for some time. As most of you know, that one of the features of the river that's very prominent, makes it important regionally and nationally, is its native mussel populations. I think the most interesting activity and results that have come out of some of the work that Teresa Newton and her group have been working on are the relationships between ammonia in the sediments, which are common in anoxic sediments or sediments with low oxygen, and the potential threat that those pose to the native mussels. This is also an important factor because of the fact that mussels are known to want to burrow into the sediments for thermal refuge when we have higher temperatures. And the interaction between these two things, the development of ammonia in the sediments and the relative overlying water temperature, has the potential for interaction. This work that they've done at UMass is part of a larger effort on the native mussel team. Again, here's a contact if you want to find out more about this, and the kind of systems models that they're looking at for mussel population biology, and the list of scientists that work there. So they're doing quite a bit of work on this topic. And this is a good point to mention that my presentation is focused on the scientists, researchers, hydrologists in the USGS that are working on these topics. In almost every case, they're working with a wide array of collaborators from Wisconsin DNR, Minnesota DNR, Fish and Wildlife Service, the River Association, the National Park Service. These are all players, and it's quite an involved group of people. And also the Science Museum, research station scientists, as well as university scientists, are involved in collaborative efforts in almost all of these projects. So I just want everyone to keep that in mind, that I'm a little bit focused here on the USGS role, but it's a small part of a collective effort. So there's also work that's going on investigating native species responses to invasive organisms. And I apologize for stopping to drink, but I have a cold, as many people do, and my throat will stop working here if I don't occasionally feel the sip, so sorry about that. The thing I wanted to point out here is that there's been some work on food web relationships for things like zebra mussels and zebra mussels' ability to persist in the lower St. Croix. That's a very interesting result. There's the issue of what happens to zebra mussels under different flow regimes. That's an area of investigation. This last project is something that Jeff Zigwide and Steve Ziegler have just begun to work on, and that is investigating how the proposed barriers that might be put in to control invasive carp species would affect large native species that are migratory, like lake sturgeon and even paddlefish. The work that Jeff and Steve are working on is to take samples, non-destructive samples from the fin rays of sturgeon, and analyze their elemental composition, trying to reconstruct their migration history. And if you were at the Research Rendezvous in October, you heard some interesting discussion about actually putting telemetry on some of these large fish, and you saw the pattern of their movement. Some of them move quite large distances, going up into the St. Croix, going down, going over into the Mississippi. So it is a real issue, and we'll see where this work leads. It's just starting. It's funded by one of the USGS Park Service partnership programs that we have. We have two, one on the biology side of our investigations, and one on the water quality side. And those programs from USGS, in conjunction with the Park Service, have funded a number of the projects I'm going to be talking about later. So as I said, the outlook here is to give you an overview, and then I'm going to provide some more in-depth detail on two of the projects. And this is one of the projects I'm going to talk about. This is work by Kathy Lee, Sarah Elliott, and Ed Furlong from our National Water Quality Lab, looking at emerging contaminants, pharmaceuticals, personal care products, and what we call other organic waste compounds that survive wastewater treatment and end up in streams, rivers, and lakes. This project is focused on sampling above and below a set number of wastewater treatment plant effluents and comparing the patterns of detections in those stream gradients to see how it relates to the effluent characterization. The eventual endpoint of this project is to examine this pattern of chemical detection of the streams and its influence on fish and crayfish populations. I'm not really going to talk much about that, but that's work that's being done in conjunction with some EPA researchers. So there's more about that in the future. But we've been doing a fair amount of work on emerging contaminants in the St. Croix, and there are a number of other projects and publications that you can find at this web link to the Minnesota Water Science Center link, or to the National Toxics. So now, sort of switching gears a little bit more into the water quality side of things. There's been a lot of work characterizing nutrient concentrations and work on estimating nutrient loads. And a recent project that was funded by Wisconsin DNR as part of the bridge mitigation funding for the Highway 36 bridge is to start measuring nutrient and sediment loads coming out of three major Wisconsin tributaries to the lower St. Croix River. That's the Apple, Willow, and Kinnickinnic rivers. The approach is to not only do kind of a standard load estimate using regression analysis, but also to measure event-based loading and to get a feel for what's coming out of these streams throughout the year. So Ben Torreson, who's with the Wisconsin Water Science Center and out of Rice Lake, and Paul Juckham out of Wisconsin Water Science Center are doing this project. And this is just a slide that Ben put together that shows, this type of event-driven loading. And I probably think you and YouTube are going to have a hard time to see this, but I hope some of you can see that this is the first sample that came off at the beginning of the event. This is in the middle of that peak in the event, and this is towards the end, and you can see how opaque those samples are. It's because of the particulates, basically the sediment particulates in this sample, and the associated nutrients on those particles in the form of phosphorus and nitrogen can be an important component of loading. So Ben has been calculating these loads, and they're available, I think, through 2013, through the Water Year 13, on the Wisconsin Water Science Center webpage. We are using these loads in our lake modeling efforts. So we are using these as part of the calibration data set for the lake model that I'll be talking about in a little more detail. And this is the lake model project, just to give you a sort of a snapshot of it. The idea is to build a biophysical model of Lake St. Croix, which involves coming up with a dynamic water balance and a dynamic, what we call a heat budget, or a dynamic temperature balance as the baseline physical parameters in the lake. Then we add in the nutrient loading, and we have a, as realistic as possible, biological response to that nutrient loading in the form of algal production and algae interacting with zooplankton, with nutrient regeneration from the zooplankton eating the algae and releasing nutrients from the algae back into the upper part of the lake, and how that process continues as you move downstream in the lake. And in Lake St. Croix, since there are a series of ever deepening pools as you go from basically Bayport to the Kinnickinnic pool, you have a interaction with the size of those basins and the amount of time that water spends in each of those individual pools as it moves down the lake. It's a very dynamic system, and as we found out in the last few years, with varying inputs, varying loading episodes, some of them coming in midsummer or early summer, as opposed to a sort of a predicted spring runoff period. Understanding that dynamic interaction is pretty important if we're going to understand how the lake might respond to our management, our planned management activities. The point I want to make is that the interaction between the research plan, if you will, or the planning of the St. Croix Basin team and the support of the Basin team in terms of ranking proposals and moving forward funding for the Wisconsin DNR funding for bridge mitigation has been very helpful to moving this process forward in terms of giving us even a more complete data set for the nutrient loads coming in from the Wisconsin trails. So I'm going to provide some more data in terms of the kinds of algae biological responses that go into this modeling as well when we get into some detail. Yes, I have a question.
Could you elaborate on the last bullet point, modeling, predicting habitat?
Yes, I can.
What was the question again? Do you mind?
Excuse me, no, I should repeat it. The question was if I would elaborate on the bottom bullet, which states the model will be able to predict oxygen and temperature dependent habitat. And what that means is the model predicts the water column distribution of temperature and oxygen. So it'll show the higher oxygen in the upper layers of the pools in the summer, and also predict the low oxygen in the bottom waters based on how much algae production there is and what happens to that algae. If that production goes down into the bottom water, it's no longer actively growing, it's decomposing, bacteria are consuming it. That consumes oxygen, and so oxygen becomes depleted in those bottom waters. The model actually includes simulations of that process. So it's actually the way we calibrate the model, having oxygen and temperature profiles in the pools at fairly frequent intervals. We aimed for every 2 weeks, is how we calibrate that physical chemical component of the model. Then, once we've calibrated the model, we can run it under different loading scenarios. We can use the same amount of water that we observe to inflow into the lake, but we can up the concentration in it, thereby upping the load and see how the lake would have responded, or try to predict how the lake would have responded. Or we can change both the amount of nutrient that's coming in and the amount of water that's coming in. That's a little more challenging to do, but it lets us run scenarios to see how the lake might be predicted to respond. This use of oxygen and temperature predictions to model habitat is something that's common to the Minnesota Sentinel Lakes program. The USGS is involved in what is termed the Super Sentinel part of that project, where we are doing these mechanistic models for a subset of the Sentinel lakes, which are set up to monitor lake response to environmental gradients, environmental change gradients, including human gradients and climatic gradients. Dan.
Richard, I think about it, this is really going to be a complex model, particularly the physical model, because it's not just a lake, it's a flowing lake. And so you have to be thinking or be able to simulate the shear that occurs with surface warm waters moving across deeper cold waters which occur. And you have to be able to simulate that over the course of a long period of time and get it all right for all the pools.
Right now, we're limited to only being really realistically to model the lake starting out with a particular set of conditions after ice out and modeling the trajectory of the lake through October, November, December. The model can be used through a full 12-month sequence. You need data on ice thickness and some other parameters, which are much harder to get just because of the weather components. So when I say that it has predictive capability, it lets us explore how the system, which is very dynamic, as you point out, might respond as we change things like reducing the loading by 20%, or changing land use in the upper part of the watershed, which will change agriculturally derived loads. And one of the long-term, we just talked about this at the Basin team meeting yesterday, one of the long-term goals here is to link this mechanistic model with the mechanistic model of the SWOT models for land use change. The models don't link directly to one another, but you can use the output for one as input to the other. And that's really one of the next steps once we get the SWOT model updated and once we get this model going. That's, if you will, the research plan. Whether or not you'll be able to do it or not, I don't know. In terms of the hydrodynamics in the model, it is excuse me here, it's laterally averaged. So it averages over a cross section on the lake or the river. But it can be divided into any depth increments you want. Usually we use 1 meter increments. And the model does actually simulate those shear forces within a segment, and then it moves the water to the next segment. And if you have too much, if the system's too variable, sometimes it's difficult to get the model to converge, and so you have to like increase the size of your segmentation. But the model was set up to work on riverine reservoirs, and it's been used in a number of systems like St. Croix. The 4 pools, I think, are another aspect of the model, of the actual Lake St. Croix that make it challenging. So, any other questions?
What model are you using?
The model is C-equal W2, and I'll have a few more things to say about it later on when I get into the detailed part. So the other thing we've been doing recently as a preliminary research effort, some of the people in the room might have been involved in this, is that we've been trying to track blue-green algae blooms in the river and in Lake St. Croix. And we have been working in partnership with the Park Service and with Marceline Mosher, who's the volunteer coordinator here at the St. Croix River Association. And she helped us set up a volunteer network. And the volunteer network included some USGS people, some Park Service people, and then some citizen monitors who were willing to go out and collect samples. And so this summer, from about the second week of July through the end of October, volunteers went out every 2 weeks and collected samples so they could be analyzed for blue-green algal biomass and then also screened for the algal toxin microcystin using a qualitative, semi-quantitative bioassay called an ELISA bioassay. And the reason we did this is because of this sort of phenomenon. The green that you see in that, the green flecks that you see in that picture are actually microcystin, or excuse me, microcystis colonies, which is one of the species of, or one genera of algae that produce microcystin. And it was quite prominent in the lake, and we found it in every site to a certain extent at different densities. We found it in every site that we sampled along the river. And our river samples, sorry I don't have a map for this, but they ran from all the way up from the Highway 70 bridge down to Osceola and down into the some of the contact recreation areas just above Stillwater, and then down into the lake, including down by Lake City where there was a big bloom near a public beach. The focus for the Park Service is trying to figure out what's going on with these blooms and what we would call areas of high probability of human contact with the water. So, and I hope to be able to come back and talk about the results of this. We're still getting the ELISA, the microcystin analyzed, and the algae are being counted. So that's kind of in progress. Any questions about that?
Just how long has that been going on?
We just did it this past summer. And one of the reasons we did that is we have a proposal, pre-proposal that we put in last year for the Park Service USGS funding opportunity, the RFP, our internal RFP. And the reviewers were complimentary, but they wanted preliminary data. They wanted to see some preliminary data, so we're using that. And I should say the Park Foundation provided us with just a little bit of seed money to sort of pay for the ELISA kits and get it going. And then, of course, the River Association volunteered their staff time to make it happen. So I think it was quite a good collaboration, and we met with Byron just yesterday to talk about moving forward with this pre-proposal again. So we hope we'll get the funding to look at this issue long term, following water masses as they move down the river with their associated algae communities, and looking at what happens to the algae as they move down the river and into the lake, how they interact with nutrients, are they actively growing, or does it all change when it gets to the lake? Because there's a lot of interest in how these blooms get initiated and what happens to them. So I have these two more detailed small presentations to talk about. I can't really see the clock. Monica, that's
12:31.
12:31. Okay. So I may go through these a little quickly, just because I want to give us time for some more questions, and the target here was to finish in about half an hour, 40 minutes. So I think we started a little late, so I think we're still good. So one of the projects I mentioned was looking at pharmaceuticals from wastewater treatment plants coming into the St. Croix River tributaries. One of the reasons these compounds are of interest is that a number of them are known to have biological effects on aquatic organisms. There's work on heart medications having effects on zooplankton. There are some effects known from antidepressants and some other drugs that are found in wastewater treatment plant effluents having effects on invertebrates. And then there's a number of compounds that have activity on animal endocrine systems, their reproductive biology, and it can disrupt their biology. It can have effect both on female and male fish, but most of the work that's been done is looking at how male fish can be what's called feminized or affected by endocrine active compounds that mimic estrogen, which is a hormone that affects humans as well as fish and a number of vertebrates. So that gives you a little bit of background about why it's of interest. And we've already talked about the St. Croix Basin and the fact that it's this premier freshwater mussel habitat in the country. The Park Service is involved because of the Wild and Scenic Rivers Act, which created the SACN Park Unit. And this work was funded by the USGS Park Service joint program within USGS. The issue really here is that there are a number of wastewater treatment plants in the basin. You can see them distributed here. And even when wastewater treatment plants are functioning perfectly as designed, these compounds are not removed. So this isn't a failure of these treatment plants. It's that they really, in many cases, just aren't designed to remove these trace organic compounds. And in many cases, the concentrations that come out are really small, and you wouldn't think that you would have to be concerned. But unfortunately, some of these compounds have biological activity at very low concentrations. And you'll see some data in a moment that this is some citations for some of the previous work that's been done. And the approach for this project was to actually move forward with sampling to fulfill this data gap and measure concentrations from 3 different sites it ended up working on 5 tributaries. That is, we sampled upstream and downstream of the effluent discharge, and then we also sampled the effluent so that we could compare the results in terms of the number of compounds and the composition of the detected compounds, and compare it with the effluent, just to try to answer the question of whether it looks like these compounds are coming from effluents, or does it look like there's a watershed contribution. The project also, I'm sorry, I should have said that the project also included this assessment of the effect of detections on fishing crayfish, and I'm not really going to talk about that yet. That data set is still being developed. So the sites were selected based on the percent of effluent contribution to base flow, and we ranged from as low as about 4% of stream flow being from effluent to 100% of stream flow being from effluent. And the other criteria were that there had to be a presence of aquatic biota, fish and crayfish, because those were the target biological, those were the biological targets of the study. And then also sites were screened for just how much estrogen active compound activity there was in the sample, just to make sure that there was some estrogenicity in those samples so that we were looking at a place where there might be a potential biological impact. So the sampling methods I've already mentioned, upstream, downstream, and from the effluent, and also we analyzed for 110 different compounds that are human use based pharmaceuticals. And that work was done in Ed Furlong's lab at the National Water Quality Laboratory in Denver, Colorado. So, what's the take? Well, 65 of those 110 compounds were detected in at least one sample, and 18 compounds were detected in at least half of the samples. The compounds that you see with an asterisk, those were found in all effluent samples. And I'm not going to go through the whole list, but basically the top compounds involve, this is a medication for treating diabetes. This is a beta blocker. Carbamazepine is an anticonvulsant. It's also prescribed for nerve pain and nerve conditions. There are a couple of blood pressure, the beta blockers here, and I think it's metoprolol is actually also used as a blood pressure medication. I have to check my notes. But tramadol is an analgesic. It's a mild opiate that's given for pain. Aciclovir is an antiviral. So the list goes on. And the point is there are a number of compounds that are coming in and they're detected. The question is, what might be their biological activity? That's the other question that this study is trying to answer. There were only a few compounds detected upstream: nicotine, cotinine, which is a nicotine degradant, and then metformin, which was the anti-diabetes or the drug that's used to treat aspects of diabetes. The important point is there were only a few compounds detected upstream, and some of the sites there were no detections upstream, and that's the next slide. This just shows you for all of the 5 treatment plants that were, for the 5 sites that were sampled, the upstream, the effluent, and the downstream number of detections, that's the number at the top of the columns, and then the total concentration of all of the detections added together. Now, in terms of environmentally relevant concentrations that might have biological impact, that starts somewhere down here for some compounds and then goes upwards. So the total concentration of these compounds taken together begins to get to the point in some of the streams where it might be an issue, but we don't understand fully how these compounds interact, or if they do interact in their biological impact. Yes?
Just a question about upstream. Does that mean that there's no treatment plants above that?
No, that does not necessarily mean that. There could be septic system discharge, there could be shallow groundwater discharge, there could be a small treatment plant that discharges. There were no major treatment plants that were upstream that I know of at these sites, but there could be other sources. And there are some ag sources of these. Some of these chemicals are used in animal husbandry, so that's the other issue. Excuse me. So the point I really want to make with this slide was that for the sample that was 100% effluent, you can see that the stream downstream looks a lot like the effluent in terms of the number of compounds that were detected. I guess the question is, well, that's true, a number of detections, but were the chemicals similar in those samples? And the next slide, whoops. The next slide shows the samples compared for those 5 sites. In each pair of bars, the first bar is the effluent, the second bar is the downstream sample. So in this sample here, which was 100% effluent, you can see that the color bars match up fairly well between the 2 samples. So it's not only that detections were common, but it actually was detecting the same compounds. That isn't always the case. At this site, which had a low percent effluent, there's only really 3 compounds, and some of the compounds become more important in terms of the percent that's found in the sample relative to the original mixture in the effluent. And this has to do with what happens to these compounds once they get in the water. Not all of them stay dissolved, some of them end up in the sediment, some are degraded, some persist for a long period of time. So there's a lot of work yet to do on understanding what happens with these compounds and what effect they might have on the bio. So, any questions on that? Oops. So I thought you might be interested in this because this is a very recent study. Sarah Elliott presented it at the Water Resources Conference, but it actually has not appeared in print yet. And so there is a scientific investigations report that will be generated out of this project and a data report. So it's kind of, well, for USGS, I guess that's kind of late breaking news. Some people have a faster time frame, but this is stuff that's just kind of coming off the press. The last thing I wanted to talk about was more detail on the lake study, and I may abbreviate this a little bit because I know some of you have seen some of these slides, and we can use it as a point of discussion. This shows the Lake St. on your well, let's see, on the left-hand side of the screen as you're facing the screen. That shows the Lower St. Croix and Lake St. Croix, the location of the 4 pools: Bayport, Troy Beach, Black Bass Pool, and then the Kinnickinnic Pool. And some interesting things about this is that based on work that was done in the late 90s, early 2000s by the Wisconsin Water Science Center and others, if you look at the volume of some of these pools, even though Troy Beach is not as deep as Black Bass or Kinnickinnic, if you look at the area of the pool that's subject to going to low oxygen, it actually has a larger surface area. And so you need the volume information to really understand what's going on with these pools. Part of that is because the lake opens up there, and it is deep enough where it has quite a bit of sediment surface that's interacting with the lake. The project I'm going to talk about, the phase 1 project, was done in collaboration with Sue Magdalene and others at the Science Museum, doing the work for the St. Croix River Association, and it was to do a better job of it was aimed at understanding how nutrient dynamics were interacting with phytoplankton and zooplankton, but also to improve our estimates of loading. Get some estimates of internal loading from those pools during the summer stratified period, but also try to understand how the algae respond to available nutrients. So we were trying to get a better handle on the mass balance, identify what nutrient was limiting. The supposition was that it was phosphorus that might limit phytoplankton growth or algae growth, and then try to develop a predictive relationship between the limiting nutrient and supply. And this really has been moved to the modeling phase of the project. Some results that have come out of this, in order to really sort of understand what's going on with loads, Jeff Ziegweit and Sue Magdalene have focused on coming up with a better estimate of discharge of basically river flow that comes into Lake St. Croix. We did that by installing a index velocity discharge gauge at Stillwater, collecting data for, I guess we're going on 3 years now? 3+ years. Yep. And then Sue and Jeff have worked very hard to do an analysis of this flow data and compare it to the historical way of calculating discharge at Stillwater, which was to calculate a expected discharge at Stillwater from the St. Croix Falls gauge. So these data that we collected as part of this project were used to evaluate previous discharge methods. And the St. Croix gauge continues to be funded this year by Wisconsin DNR funds from the bridge mitigation projects. It's really critical to our understanding of what's going on in Lake St. Croix. And then we did phytoplankton and bioassays and zooplankton grazing bioassays to try to understand how phytoplankton growth rates respond to the available nutrients in the lake and how they would respond to nutrient additions in an attempt to document nutrient limitation values. Okay.
We've got 5 more minutes.
Okay. Well, then, oops, I'm sorry. I started to go the wrong way. Okay. So this was the index velocity. And what I wanted to show here is just that the reason we needed to do this is if you compare can you see, Sue, can you see the red, the green, and the black lines back there?
Yeah.
Okay, well, the old method of estimating was shown in red from this period of record, about a year's worth of record, shows that under high flow events, the old method was overestimating discharge, and under low base flow conditions, it was underestimating discharge. So there was room for improvement. And this is just one analytical approach, which was a one-day lag. There's a scientific investigation report that will be coming out very shortly, we hope, next month or two. And that goes into this into great detail. But this kind of data collection was critical to being able to move forward with the lake water quality model, because you need to know what the loading is like. Whoops. So the bioassays were used to develop estimates of algal growth rates, and this just shows pool 1, 2, 3, and 4, all the algal growth rate bioassay work that was done for controls or ambient growth rates. So these are the growth rates observed in the lake, and then what happens when you add nitrogen, phosphorus, or nitrogen and phosphorus together, and you can see from these groups of peaks that when you add more nutrients, the algae tend to respond. That tells you that they're not nutrient saturated. But what's more interesting is to look at the control growth rates relative to, let's say, phosphorus addition or nitrogen addition. That tells you where you have a pattern of nutrient limitation. The thing I'd like you to take away from this is that there's variation from year to year. So the pattern that you see in June and October in 2011 is not matched up with the pattern you see in June and late August in 2012. And this is coming out of some other kinds of analysis that are being done on water residence time and loads, that the environment that the algae find themselves in changes dramatically. And so the algae response to available nutrients is dynamic. Suggests you're going to need a dynamic assessment tool in order to track that kind of change. And I'm going to skip this part where we actually looked at, we did sediment core incubations just to get estimates of how much nutrient was coming off the sediments in these pools, because we wanted to understand how that internal supply of nutrients might affect these summer dynamics and growth rates. If you look at this early season in April, before you really have stratification, there's very little nutrient release from sediment cores when they're incubated in the lab. But if you go to the period of anoxia, and these sediments were anoxic when they were incubated, we get much greater rates of nutrient release, and we can use those estimates to look at how much nutrient we expect to be accumulating in the hypolimnion that might become available for growth in the upper layers of the lake. We do that by taking those release rates and estimating from our profile data how much of the basin, here shown, of the base sediments shown in red, were at low oxygen or anoxic, and then we use that to calculate seasonal internal loading that's coming into the pools. So based on the release rates we saw, you can look at very little release coming on early in the year, because there's no anoxia, no low oxygen water at the bottom of the lake. But as the lake inflows decrease and the lake pools become more stratified, we tend to get greater loads. And our results shown here are within the range of what was used or observed in the previous bathtub model. So we've actually come up with a pretty good estimate of what's going on in total loads I think the other thing that's important to look at is that depending on whether you have a dry year or a wet year, there's quite a bit of variation in the amount of internal loading you can get. Because in the dry year, you're going to have a longer period of stratification, more column stability, larger amount of anoxic sediments, and more nutrient basically being released. So that's the kind of data collection that it's gone on and we continue to do with our profile data, estimating the extent of low oxygen or no oxygen sediments to calculate internal loading. And we're using that in a mechanistic water quality model. And I think I'm going to stop here because we're running up on time, but that model, we're running out of time, but that model incorporates these observations into a dynamic framework. So it takes into consideration the dynamic inflows, changes in meteorological conditions, and the changes in water and nutrient residence on the lake. So that's well said. Yeah.
A question about your last slide. The first bullet point determines why current levels of phosphorus reduction have not reduced seasonal planktonic algal biomass. So it seems to me that assumes that there are some reductions going on. Tell me more. I didn't realize that it was going down.
That comment comes mostly from a paper by Brenda Lorenzo-Francois and a number of other authors that did observe. Oh, thank you, Sue. I was trying to think, okay, who in the room was on that paper?
Brenda, me, and Kent.
There you go. That and here now I'm in danger because I'm going to paraphrase someone's work that's in the room. But the idea was that there were some observations of indications that there had been some reduction in loads, and yet what was happening is that although phosphorus concentrations were declining a little bit, there didn't seem to be a corresponding decrease in algal biomass as measured by chlorophyll A. So the question is, why is that? But you ask a very good question. Is that a long-term pattern, first of all? So is it really a problem? Yeah, only with monitoring will you know that.
I would back it up further and say why are there reductions? What's causing the reduction?
Well,
What's behind it?
land, everything we've done in terms of land use improvements, mostly wastewater treatment.
Okay. Better wastewater treatment.
Yeah. So now we're facing non-point source reductions to get the rest of it.
But if the subcommittees of the basin team are working on accounting for other sources and tracking how best management practices as they're implemented are affecting loads. There's quite a bit of activity, in both states on trying to quantify what's going on in terms of load reduction associated with different kinds of land use. So the idea is that as we track empirically what's coming into the river and if that changes, but also as we track what we think management is achieving, what effect will it have on the lake? Because one of the questions that needs to be asked or that needs to be answered is, we've set a target for reduction of loads. Is that going to get us where we want to be with the lake? Because other factors are changing. Mm-hmm. Inflow patterns appear to be changing. Temperatures, average water temperature appears to be changing. Mm-hmm. These things can interact to create a different pattern of oxygen depletion in those bottom water, might cause more internal loading. So even though you're reducing external loads, you might have a response in internal loading. Midsummer flushing, if we really have established a pattern where we're going to get big inflows in June and July, maybe that washes more nutrients out and the lake doesn't stratify as much and maybe you don't get as much internal loading. This is a very dynamic system and it, physically complex, and you need a dynamic tool to try to answer some of those questions. And actually to get at some basic science questions.
Given all of that, is it a fair assumption that the science points to saying we've got it as far as we can with wastewater treatment to reduce the bad stuff going in, and so now non-point is good?
The time of year, so I work with the wastewater treatment plants in Wisconsin, and when we set about this, we, first, the baseline for this was in 1990s, before we required phosphorus removal at our larger treatment plants. So since that time, Wisconsin's reduced the phosphorus from point sources by 60%.
50?
Yeah, and now we've implemented new phosphorus rules that for some facilities, they'll keep the limits they had. It's all about assimilation. If you're a small plant on a big stream, you get a limit you can easily meet with technology. But our new phosphorus stream standards were passed in 2010. Some facilities are going to get extremely tight limits, way lower than the TMDL requires and way lower than the states in the past. In fact, limits that are lower than the technology can be reached. So we will still see some reduction in Wisconsin from our point sources, selected ones, small ones, Siren, Clayton, you could probably name off the ones in Wisconsin. They have a long time frame to meet that because it's going to be difficult and it's going to be expensive. If they can't do it through treatment, they could also trade and take out a higher ratio of pounds from the landscape, whether it's stormwater, whether it's trading with an agricultural producer. So we'll see some, but we're getting down to, we're talking instead of 60% of the gross quantity, we're fine-tuning a smaller number there. So we still will see some reduction.
Okay.
Right, we saw a big reduction in
It's
St.
like
Croix Falls.
Bluegills.
Kathy worked hard with Joel, got the city administrator for several years to get a new plant online, and from a 4% down below 1%.
Yeah, 4 parts per million down
Oh,
to
4
about
parts,
1.
yeah.
Brantford's one of the other larger contributors. It's similar, going down from like 6 to 9.
Well, I'm sorry to dominate the questions, but my interest is in trying to find bullet points that would sell with the general public and particularly potential donors for the River Association, because clean water is a big deal for them. And I've got to come up with, 5 or 6 key sentences that says here's what's going on, here's what the opportunity is. That's a complicated model.
I can tell you that from just a very small sample size, when we were doing the Algae Blooms sampling, Ken Johnson at council, some others in the room, and myself, we got phone calls from citizens, wanting to know about those microcystis blooms, what was going on.
Yeah.
There were some pretty big accumulations of biomass as the algae tend to float to the surface and blow into a different shore, and it can be there for a few days or even longer, or sometimes it's there one day and gone the next, and people wanted to know what it was, get out, wanted, was anybody sampling, what does it look like? And that is connecting with people in the basin. So understanding what's helping to drive those blooms, and whether it's related to the nutrient economy, which probably plays some role, but also to the pattern of stratification and water temperature and some other factors, I think is a public concern. And the other thing to keep in mind is it is a rapidly changing basin. It is urbanizing in the lower part. It is got a large recreation component footprint, and there's a lot of land use change that's going on, driven by that urbanization, but also by agricultural market forces. So it's a dynamic system, and we need dynamic goals to really keep on top of it.
It's all helpful. Thank you.
On your aquatic invasive species slide, I was amazed to see that fish are eating 72 to 94% of zebra mussel biomass. Do you know which fish in particular?
No, I don't.
It's not
That's
carp.
that's not my bullet.
Okay.
But I will find out that information. This is where I would like to put in a pitch for one of my favorite fish, which is bluegills, because from the work that's been done out of Peter Sorensen's lab, where they've been doing carp removal and restoration in some small, really small lakes, large ponds in the metropolitan area in Minneapolis-St. Paul suburbs. Once you've reestablished bluegill populations in those ponds, they eat all the common carp eggs. So I just, it's one of, and it's like, boy, that was an aha moment, because I was like, whoa,
It's a native.
because I work a lot with sunfish and their response to emerging contaminants, and I like bluegill sunfish. They're
I thought it
tenacious
was,
and tough.
drum and.
Well, drum and freshwater sheepshead drum
Yeah.
actually eat bivalves, and I would assume they eat zebra mussels.
No.
And
Some of the red horse.
maybe
Yeah.
there's a potential biological control angle. If you can raise enough sheepshead, they get big and they produce a lot of progeny. So
And
and they're native. Oh, I'm sorry, Gary. Yeah, go ahead.
Just back to the emerging contaminants for a minute.
Sure.
Good research here, and I know Don did some earlier too. Is the rest of the country doing the same thing? And do you see any type of changes coming in our wastewater treatment plants to deal with that?
Well, I will say this, that the LCCMR funded a project through the Environmental and Natural Resource Trust Fund in Minnesota to Paige Novak, who's a professor in engineering at the University of Minnesota, and some of her colleagues to look at different kinds of treatment of the project in detail, but it is trying to look at treatment options and how it affects the suite of compounds that come out of treatment facility, and also looking at the biological impact on fish populations. And that piece of the work, I believe, is being done by Heiko Schoenfuss at St. Cloud State University. Heiko and I have collaborated on emerging contaminants in lakes and trying to look at how bluegill sunfish and benthic minnows respond. So that work is out there. Some work that was done on another class of contaminants has looked at like water residence time in treatment plants. And if you lengthen the water residence time of treatment, sometimes it releases or it removes more of these trace organic compounds. They end up in the sludge, and then you have another pathway that you have to be concerned about. I would suggest anyone who's really interested in this topic to go to the USGS National Toxics Program page, because there's quite a bit of work that's going on in USGS on this issue nationally, and there'll be links to other work. Kathy Lee, who was one of the principal investigators on that study, is now an assistant director of that program at the national level. Kathy's still here in the state, but she's taken on more responsibility, and I know that's a big interest. For example, one of the things they're looking at is these compounds coming out of leachate in landfill systems.
That's a big issue.
Yeah,
We say they're all lined and the leachate is collected, and where's it go?
Well,
It's a
and
wastewater
some of
treatment
it's
plant.
treated and some of it isn't. But I think in all the cases where they sampled, it was a system that had a collection process that allowed them to pull a sample of leachate from the landfill. And I made the point about concentration. If you look at the concentration in some of those samples of these compounds that are held in common, those concentrations are much higher than the concentrations we see here. Anyway, so nationally, yes, there's awareness, but it's a lot of compounds, and I think there's a desire to understand how compounds respond, how biota respond to the compounds before they move in any direction to try to maybe regulate those compounds. But you asked about treatment.
One quick follow-up. Do you have
Sure.
any idea how much atrazine is in our river?
I don't have those data available to me. Atrazine is part of the other wastewater compound schedule that we run, and we probably have some data on that, but I don't have it ready to share with you today. I'd be happy to try to pull it together and see what.
Thanks.
Over here, Evan.
Oh, I just wanted to echo what Richard was saying about the number of calls from the public that we get in regards to algae blooms. Public's really concerned about algae blooms, not just the blue-green algae, but I learned it's what it is. It's benthic algae that forms filamentous mats, and they grow on the underside of rocks. And then in midsummer, they break off and float down the river. And every year in midsummer, when it calls, there's poop floating down the river because that's what this looks like. It's poop. Yeah,
Yeah,
it's
it's,
brown.
it's usually a filamentous green. Sometimes,
Yeah.
in some cases, it's diatoms, but I don't, not in this case. It's filamentous green algae, and they, as part of their development, they kind of float up to the top. They senesce, they
Yeah.
die off.
And they seem to be increasing, and people will call and say, I have, fished on the St. Croix for, 20 years, or I've lived on the St. Croix for 25 years, and I've never seen anything like it. There was this map that extended all the way from, the Minnesota bank to the Wisconsin bank on the Indian floating through. So, I mean, if you're looking for something that people are really are worried about, and help
Well,
fund them.
I will say this, that Byron and I are, and others are resubmitting our proposal to try to look at some of these bloom dynamics of the, not just, I mean, it's focused on Bluegreen algae, but one of the things we've talked about is putting continuous monitors at some of these contact recreation points, so that that data can be used to figure out when it looks like we're getting a bloom dynamic at that location.
Yeah, that'd be great. And I know the Turk Service has a proposal in Copper too that may be funded in the next 2 or 3 years, but people want answers now. They don't want the answers 3 or 4 years from now.
Is that what involves my family?
Yeah, with Brenda.
Yeah.
Brenda is on that project.
Right. It's still with the child.
Yes,
Some of
yeah,
the-
the cause and extent of
Yeah.
algae, nuisance algae mats, I think it's called.
Mark is developing a new Center for Harmful Algae Response in Minnesota, or CHARMS lab at the research station.
We actually grabbed some of the stuff and put it down in there to tell us what it was, so
Yeah.
thank you, that was very helpful.
So, we have a question?
Well,
Do you have time?
if you don't mind, I'd like to wrap it up for the
Yeah, okay.
YouTube people.
Sure.
The
Thanks,
rest of
folks.
you here can stay and chat, but Don, one last thing to say.
Yeah, I would just like to thank Richard for joining us here today for the water forum. Thank you very much. And also stay tuned. In February, we hope to have another forum around mid-February. Thank you.